So I'm very pleased to be here. Thanks for the invitation. I've been fascinated what you've all achieved over the last several years, so I hope I can give a little contribution of where we might take some of these more standardized approaches. So I'm going to talk about our moves towards developing stem cell-based drug screening, and I'm going to use cardiovascular diseases as a kind of example, but of course you could read into this many other kinds of diseases, of disease modeling. But I would just put some points of warning, given that you might be moving to the second round of this, of the kind of shortcomings we have in these models. So just to get us all on the same page, it's 9 o'clock in the morning on a Friday. We have embryonic stem cells. We have had them for a long time. We know they divide in more or less indefinitely. We know that they can form all cells of the body. We can get them from diseased embryos, so by prenatal genetic diagnosis. And from these, we know that these are the most serious disease because they will be derived from IVF. They've been around for a long time. The disadvantage of these cells, though, of course, is you've never seen the individual from whom they come. So we don't know what the phenotype of that individual would have been if they have a disease. We don't know how severe it would have been, and we don't know what the age of onset would be. Now, because of the ethical issues with embryonic stem cells, there was a sort of enormous revival of adult stem cell research. 30 years, 40 years ago, hematopoietic stem cells were discovered. They're routine in the clinic for bone marrow transplantation. But I think Hans Clevers has been really instrumental in telling us about another type of stem cell in adult tissue. So every tissue that can repair itself appears to have a stem cell population. Now that has certain advantages. So these stem cells are rather mature. So the embryonic stem cells always give you rather immature derivatives at the present time. But adult stem cells are fairly mature, so they will look like the tissue they come from. They're very close to the real thing. And that's most evident in, let's say, the intestinal stem cells, the gut stem cells, colon, stomach, lung, liver. All the gastrointestinal tract appear to have this stem cell population that would also grow indefinitely and grows in the form of organoids. The downside of these is they will only form the cell types of the tissue from which they come. So they will not form the stromal component of the tissue, and they will not form the blood vessels. And in some cases, diseases are caused by this dialogue between the stroma and the vascular cells. So you will not be able to mimic that kind of model, the more complex models. And that's where iPS cells, of course, fill a very important role. Apart from addressing the ethical issues, they can be derived from any individual at any age, which means we know much about the disease as it manifests in the patient. And we can make something like embryonic stem cells. Again, they will make the immature component of the tissue, but we can make all of the components of the tissue. And we focus primarily on the iPS cell as disease models, simply because we work with diseases which have more cellular components than just the target cell of interest. So this is a sort of overview here. Like I said, we have the iPS cells here that can be derived. Of course, there's been a lot of interest in using these cells for therapy. But this is really, in general, fairly far down the line. We think macular degeneration is going to go ahead. There are about 80 patients now included in trials throughout the world, and some of them look fairly successful. But it is a challenging operation. It looks like Parkinson disease, transplanting dopaminergic neurons, is going into the clinic perhaps even as early as next year. They work very hard to get the right sort of dopaminergic neurons. It's looking extremely promising. Again, in part because the transplantation has already been done with fetal cells more than 20 years ago in Sweden. And we know from the autopsies of those people that later died of other reasons that those cells, the right cells, remain integrated in the brain over that long period. And a third condition is probably or possibly pancreas cells will go in for severe forms of diabetes. But many of the other applications are really quite far down the line, in my personal opinion. So in our short-term applications, we're looking towards disease modeling, pharmacological screening. There are shortcomings to animal models. The brain of the mouse is quite different from humans. The mouse doesn't have all components of the brain that a human does. There's different structure of the stomach. There's a completely different structure of the immune system. The heart behaves completely differently. I'll get back to that in more detail. But it's important to remember that the immune system is a trigger for many kinds of diseases. So that if you don't have the right immune system, you may not be able to model diseases and find cures. And an example is they've worked with a mouse model for sepsis for more than 20 years, but there's still no cure. So pharmacological screening on human cells can be of great value, but also toxicity tests. A reasonable number of drugs are withdrawn from the market because of toxic side effects, and if we have the right human models, we may be able to pick these up. So these are some of the things I'm going to cover. So what are the main problems? There are no drugs for many chronic diseases. Existing drugs don't work in all patients, so people find it quite common. You go to the doctor, it gives you a drug, come back in 3 weeks if it doesn't work, and a large number of patients do come back, it hasn't worked, try something else. Would we be able to get more directed and accurate prescription of medicines if we had the right models? And as I said, drug side effects can be toxic. They're actually the 4th leading cause of death. That's something you should really think about. You know, not cancer. Cancer, of course, and cardiovascular diseases are high up, but this is number 4. That's very high. And why is this? Well, there's poor insight to human disease mechanisms. We know a lot about mouse disease mechanisms, but less about human. There's a lack of personalized treatment prediction, and animals are often poorly predictive of humans. And as I said, there are several examples. The heart, the immune system, the brain, the reproductive system, and the stomach are quite different in most animal models. And just to show how iPS models have developed over time, this is a summary we made for the 10th anniversary of iPS cells on the number of papers claiming to describe iPS disease models. And these are all for genetic forms of disease. So the first paper reporting iPS cells from patients was from George Daly's lab in 2008. And you can see this explosion of papers up to 2016, we've looked here, in different areas. And we split them up into neural diseases, cardiac diseases. Some were kind of organoid sort of models, endoderm, mesoderm, and you can see this has grown year by year, and it just shows the huge amount of interest in this area. So, but there are problems, so why don't we using this every day, all the time, and why aren't pharma using it? We know there's line-to-line variability. We don't know the cause of this, but there is. Directed differentiation to the required cell types, that's still rather in its infancy. And one of the exciting areas have been the many companies coming into this area providing you with kits. So at some point in the future, making any cell type will be like making a cake mix. Just mix it in and it will work. So this is an area of very rapid development, which has been an emancipation for many people coming into the area new, and particularly because of all these protocols being serum-free media. So there's not the batch-to-batch variability as we saw with serum-based protocols. The maturation of the differentiated cells has remained an issue. It seems to be soluble by waiting a long time for neural cells, so you can get beautiful neural maturation to some extent, but you have to wait maybe a year before you've really got them. But other cell types, they're immature. So if we're modeling diseases that manifest in adults or old age, we can't capture all aspects of that. Some aspects we can, but not all. And genetic stability. So I think Peter Andrews' group has been pioneering in this area, but there are, of course, many other groups. Wisconsin's been looking at it, but many people have been looking at genetic stability. So the solutions could be in defined culture conditions. Maybe we can get maturation and better disease models by making complex multicellular engineered tissues, so microtissues in some formats, and physical constraints. So a tissue is not sitting there passively on a plastic culture dish. It's on a soft substrate, it may be on a slightly harder substrate, it'll be subject to flow, stretch, strain, and stress. This is the things that are often missing. And particularly for the heart, it undergoes cyclic stretch and strain. Could we solve this by electrical or optogenetic pacing over long periods? The blood vessels, there are 60,000 kilometers of blood vessels in your body. Almost every cell touches a blood vessel. But fluid flow through those vessels may affect how the endothelial cells behave, and they are very important. Different energy sources. When you're born, you switch from using glucose to fatty acids the moment you drink your mother's milk. There's a very important change of energy source at birth. Maybe that will help us move forward, because all of the media we use now are high glucose, right? They're like drinking sugar. And maybe certain hormones that kick in at birth will help us in this area. So these are just sort of food for thought, and some of them have been important. Sorry. So how do we get cells to undergo directed differentiation? Basically, we use the principles of developmental biology, and I won't go into all the details, but basically we have to make cells first form mesoderm, ectoderm, or endoderm. And in our case, we want to make heart cells and vessel cells, so we need to go to mesoderm. Then you add another bunch of growth factors or inhibit processes, and you end up getting beating cardiomyocytes. Takes the first beating appears after about a week, and then about 12 days you have beautiful beating cells. If you want to make vascular cells, basically you go through similar steps. You first need to make them form mesoderm, and you add a bunch of factors, and then you add the crucial growth factor, which is VEGF, vascular endothelial growth factor. And what you see as time goes by is you get very epithelial-looking cultures. Many of these cells are CD31 or PCAM positive. That's a marker of endothelial cells. And you can fish out those cells here. So they express E-cadherin, PCAM, CD31, and von Willebrand factor. Now, this is a very typical marker of endothelial cells. Its formation here is rather different from adult cells, but nevertheless it expresses it. What you get out of this differentiation protocol is a CD31 negative population, and this is quite an interesting population, because it looks like something we call pericytes. And pericytes are the cells that surround vessels, right? So they're the cells normally adjacent to the endothelial cells, and if they're very close to the endothelial cells, they will receive signals from the endothelial cells. So if we first isolate these CD31 negative cells, they don't express these two markers, calponin of SM22, for smooth muscle cells. They're negative. But if we add in growth factors that are secreted by endothelial cells, so TGF-beta and PDGF, they switch on these markers. So just in a dish, we can get a pericyte to vascular smooth muscle cell switch. And people who are working on what we call MSCs, I won't define that term, probably are working with this type of cell, something that comes from the stromal component, and it's a rather sort of mesodermal stromal cell. So I was just going to illustrate the importance of co-cultures. So we have these CD31 positive and negative cells. You can see them here. The green ones here are the endothelial cells, and these are MSCs, pericytes, or vascular smooth muscle. They all seem to be a sort of conglomerate of the same thing, and you can mix these cells up together. And this is basically what you see. So the green ones, as I said, are the endothelial cells. They form in 2D this kind of network of vessel-like structures. And the blue cells are the CD31 negative cells, so most of them here do not express SM22, but the ones very close to the green cells switch on vascular smooth muscle markers like SM22. So they will be receiving the TGF-beta and PDGF from these cells, and will be recruiting the pericytes to make this switch. So this is kind of making a very nice vascular network. And I illustrate this point by looking at how this mechanism, what is the underlying mechanism of this recruitment, and it's based on Notch signaling. So Notch is a signaling pathway that mediates the interaction between pericytes and endothelial cells in the body. And if you inhibit Notch signaling in this system, this is what you see. So this is DAPT, which inhibits Notch signaling, and you see there are very few of these red cells. So these pericytes haven't undergone interaction with the endothelial cells. They are too far away, and just the odd one that touches makes this switch. So why is this important? Well, there's actually a disease, a genetic disease called CADASIL, which is caused by a mutation in Notch3. And this manifests as a vascular condition in which the vessels hemorrhage. Now, if we hadn't had these two cell types present, we would never be able to model this disease. If you were just looking for a phenotype in the endothelial cells, you would see nothing. So this is illustrating the point, you may need multiple cell types to see a disease. And this actually is an overview, basically, of the blood-brain barrier, but it could be any sort of vascular condition. This is vascular smooth muscle cells, and these are all the surface molecules and secreted factors associated with the vascular smooth muscle cell. These are endothelial cells, and in this case, this would be astrocytes. And if you look, mutations of any of these genes cause all of these hereditary conditions. So if you're trying to model any of these conditions, you're going to need these three components, right? You won't see anything otherwise. So this is why directed differentiation protocols to get pure populations of the right cells and that you remix them in the ratio you need in the right conditions is important in when we go forward in making vascular disease, or any disease come to that. And it's also important to realize that it's not just getting a pericyte or an endothelial cell. Each organ has its own type of pericytes. There are at least 7 developmental origins for pericytes. And each organ has its own type of endothelial cell. Now you can see here, these are 2 vessels, and these are 2 vessels that are damaged in diabetes. One is for the kidney and one is for the eye. And you can see these vessels are completely different, yet the diabetes will cause fibrosis in these vessels and cause blindness or kidney failure. So if you're trying to mimic this condition, you have to have the right endothelial cells for the kidney or the right endothelial cells for the eye and the right pericytes. So this again asks for profound knowledge of development or the right sort of kit from the companies. So, and endothelial cells themselves are very plastic, so it might not be development that turns them into the right cell type. It might be the cell adjacent to them. So they know where they are and they can switch identity. So let's get to the heart, which is my particular area of interest. As I said, there are multiple cell types in most organs, none less the heart. So the adult heart, so this is a picture of the heart. It has different chambers. Each of the chambers has different sorts of cardiomyocytes in it. There are multiple sorts of cardiomyocytes in the heart. There are pacemaker, atrial, ventricular, conduction system. But in the adult heart, cardiomyocytes are only 30% of the organ. The rest are non-cardiomyocytes. They're either epicardial-derived fibroblasts or cardiac fibroblasts, or they're endothelial cells. And it's important that we can make all of these cell types if we're truly going to make proper heart tissue. And the dialogue between the endothelial cells and the cardiomyocytes is really important. So they form the myocardial vasculature, and this supplies oxygen, but also free fatty acids to the cardiomyocytes. They actually feed the cardiomyocytes. And they're stimulated to form fatty acids by VEGF secreted by the cardiomyocyte. So there's this dialogue between the cells, this release of paracrine factors regulates cardiomyocyte metabolism, survival, and contractile force. So we have to make these more complex systems. And so one of the efforts we've been making is to try and make the right sort of endothelial cells for each organ. And what we've developed fairly recently is a system where we can co-differentiate cardiomyocytes and endothelial cells from a single starting point, so a single cardiac progenitor. And that depends on adding different growth factors, and we can get either pure populations of cardiomyocytes or endothelial cells, or a mixture of the two. Then we can mix them back in the right ratios, and this is what these microtissues look like when we mix them back. So the red cells here are the endothelial cells, and the green cells, they have transgenic reporters have this green marker here. It's actually marking the NKX2.5 locus. And you can see what this differentiation protocol looks like. So GFP is marking NKX2.5, a cardiac transcription factor, And you can see we get quite a lot of cardiomyocytes. But out of this protocol, we get NKX 2.5 negative CD31 positive endothelial cells. And when we look at the properties of these cells, this is just a PCR, and we compare them with, let's say, human aortic endothelial cells. These are primary cells, HUVECs, which are people's most favorite endothelial cell, which is actually really not a very good endothelial cell, and human blood, dermal blood endothelial cells. And what you can see here, if you look at the gene expression patterns, and you compare this with true primary cardiac endothelial cells, our endothelial cells from the iPS cells resemble, in terms of their gene expression patterns, very much more the cardiac endothelial cells. And actually, these endothelial cells are really endothelial cells, but they express cardiac transcription factors. And that's true for most organs. So they will express transcription factors of the organ to which they belong, which is important to know. So this is how these micro-tissues beat. I have to be patient. Oh, yeah, I'm too impatient. So we can do sort of quantitative analysis of how these different micro-tissues react. So one of the clues is you need quantitative measures. You can't just look at it and say, oh, it looks different, like it's beating differently. You've got to measure something. So we measure force of contraction, action potentials, calcium transients, and many other properties of these cells. And this sort of vector analysis, optical flow it's called, gives you one sort of measure. Now, so as I said, this is how basically development of disease models has evolved. We started, when iPS cells were first around, we started with making aggregates of cells, we call them embryonic bodies, and you get mixed populations of cells. These actually were not too bad, except you didn't have much control over the other cell types were there if they were not your cell type of interest. So in most embryonic bodies have an endodermal component in them, so visceral endoderm-like cells, and you don't necessarily want that if you're making a model for the heart. You don't at all. So then there was a lot of efforts put into making monotypic cultures. Everybody wanted pure populations. So if you had a differentiation protocol, they asked, what percentage of cells do you get? So it went for the heart from about 1% in the very first protocols. Most people can now get about 80 to 90% cardiomyocytes in the differentiation protocol. But as I said, it was then realized that using monotypic cultures may not be the way to go. So people are now making from iPS cells and embryonic stem cells really organoid-like structures, and these are absolutely beautiful. You may have seen the brain organoids and the gut organoids and the lung organoids coming from iPS cells. They have not only the epithelial components of the organ, but also the stromal components, and they're also looking much more mature with these other cell types than just the monotypic cultures. And of course, as time's gone on, people have started towards making engineered tissues and even more complex organs on chip formats to include biophysical parameters that could be changed. And that's where my group is heading. And you can see this reflected in the literature again. It's always good to make a review now and again. And we reviewed this area in 2016. And if you look at the publications, so in this period, you can see, for example, around 2011, most of the studies were being done on monotypic cultures. Did I see my phenotype in my pure populations of cell type X? But of late, you can see people are moving towards heterotypic cultures, and now much more to micro patterns, microfluidic structures with microarchitecture or extracellular matrix. So you can see they're divided now in disease modeling, about a third over these different areas. And I think that's something to realize where the field's going. Now, in terms of the heart, I'll focus here a little bit on that, because then I can go into more depth of actually how you might tackle it, but it could be another tissue. So you have to have a readout of your cell identity, and preferably that's not simply a surface marker or a transcription factor, preferably it's something functional. Now for the heart, and also for neurons, it's quite easy. We do patch clamp electrophysiology and look at the action potential, it's called. So these are these electrical pulses. In the left ventricle, you get this sort of shape. So I can say that's a ventricular cell because of the shape of the action potential. And here, this is the atrium, And you can see these are atrial type cells and these are pacemaker type cells. So in our first differentiation protocols, without doing anything particular, 85% of our cardiomyocytes were ventricular-like. So when we did the electrophysiology, they all look like this, the majority. There are about 15% with this type of action potential, but less than 1% were pacemaker-like cells. And this spontaneous beating you see is typical of a fetal cell. An adult cardiomyocyte does not beat by itself without a pacemaker. So anything you see beating, you can claim it's mature, but it isn't, because it's beating by itself, because there will be very few pacemaker-like cells in there. So we wanted to know how we could direct differentiation to these cell types, and basically we used the principles of development. So this is an overview of many years' work. So you go stepwise through all the different stages of differentiation, through nascent mesoderm, expressing these markers, early cardiac mesoderm, heart field-specific progenitors, and finally making cardiomyocytes. Each of these stages is marked by a set of transcription factors, and we spent several years in an EU project called Plurimes, making reporter lines for each of these. So we have a whole bunch of reporters, sometimes two-color reporters, so that we can optimize each of these steps. And this has allowed us also to fish out these cells at different stages and identify cell surface markers, because you don't want to make an IPS reporter line for every single disease. It's too much work. You want to be able to select out the cells on the basis of cell surface markers, and it turns out that SERPA and VCAM associate, for example, very closely with NKX 2.5, so you can fish those out of mixed populations, even from a non-reporter line. By using a trick in which we've put a DOX-inducible MYC into the undifferentiated cells, we've been able to make the cardiac progenitors proliferate. So we were very jealous of the people working on neural disease, because their neural progenitors, they proliferate by themselves, which is most frustrating because we've never been successful in doing that with cardiac progenitors. But by doing this trick, we can get them through 70 population doublings and then take away the doxycycline and make them differentiate to what we want. So it's a way of expanding these cells. If we add in retinoic acid at this stage, we get them to form atrial cells. So we first added retinoic acid several years ago, but Gordon Keller's got a recent paper where he added retinol, and that has a much longer half-life in your culture, and his process is rather more efficient than ours. We get about 60 or 70% atrial cells. I think he can go much higher. And what we can also do by starting with these cardiac progenitors, we can add another set of different growth factors, and we can get these to form pacemaker cells. And these cells, we identified podoplanin as a cell surface marker, so we can isolate these pacemaker cells. So this gives us a whole repertoire of diseases we can now look at, and drugs, because cardiac arrhythmia is often caused by an effect on the atrial cells. We can now get these populations of cells and are now testing drugs with some companies that could affect the atrial cells. And it's really nice. They have no effect on the ventricular cells, but a lot of effect on the atrial cells. And the same true, we can now model pacemaker disease of the conduction system. So why are mouse and human hearts different? Now, they're certainly smaller mouse hearts, about the size, less than the size of a peanut. They beat faster. A mouse heart beats at 500 times a minute, human heart 60. If a cat runs after a mouse, it still beats at 500 times a minute. Stress responses are different. If a tiger runs after you, I bet it doesn't stay at 60 times a minute. So stress responses are different, and this is because ion channels are not used in the same way. Now, this may be a little bit complicated for those of you not into ion channels, but basically the length of the arrows tells you how much current is carried by that particular ion channel across the plasma membrane. So this one here, INA, is the sodium current. You can see this arrow is this length. And if I go to the mouse, this arrow is the same length. So if I have a mutation that affects this channel or a drug, the response will be seen in the mouse and in the human. So it's a fine. But look at this channel here, this potassium channel. Now, this is a really important potassium channel. It's the cause of toxic effects on the heart in many cases. It causes what we call long QT syndrome. This is what this stands for. It means your heart rate becomes very abnormal. Now, in humans, it's very big, and in mice, it's tiny. So a drug hitting that channel or a mutation hitting that channel, you will see nothing at all in mice. So this is why this is not a good model. And there are many other reasons, but I just illustrate that one as being the easiest. So this is what an ECG, an electrocardiogram, looks like in humans. This is what it looks like in mouse. It's quite different. If you put them on the same scale, you can see this difference in heart rate very, very easily. And what we can do is, with our pluripotent stem cell cardiomyocytes, we can measure a sort of ECG. We put them on these microelectrodes, and they're sitting there beating away, and we measure an external field potential. And this is what it normally looks like. So this is very regular, beating away very happily. Put in a drug that's known to cause arrhythmias, and it goes all over the place. So these sort of diagrams you can quantify. You can quantify these little bumps here. They're called early after depolarizations, and you can measure the distance between the peaks. So this gives you a sort of readout of what this particular drug's doing. So if we look at disease modeling with some of the type of measures we're doing, one of the points I'd like to illustrate and emphasize is the importance of using isogenic pairs. So we derived cell lines, iPS lines, from a patient with a long QT syndrome, and we repaired that line by homologous recombination. So this gave us a set of isogenic pairs. And we also took a human embryonic stem cell line in which the NKX 2.5 locus had been targeted with GFP. And then we introduced the same mutation into this pair. So we've got two isogenic pairs. So the only difference between these lines is this particular mutation, as far as we know, because we formally don't know whether there's anything else wrong with the iPS cells. So this is why this was quite a nice control, just to have the one mutation here. So you can ask the question, is the mutation specific for causing the phenotype? And basically this is what we see. So if we look, this is, when we did this study, we've looked at the long QT line, the corrected and the diseased, and what you could see was when we corrected the line, there was this shortening of the action potential by patch clamp electrophysiology. And this is the human embryonic stem cell line, this is the normal, add in the mutation, and there's an increase. So the increase is about the same in both. So we can conclude probably there's no other mutation in the iPS cell, therefore probably no other mutation in the patient. And you can see these quantified here. But one of the things we found quite worrying when we did this study was the absolute values of this here compared with iPS cells and ES cells. So we asked the question, are iPS cells actually the same as ES cell-derived cardiomyocytes or not? So that was a little worrying. And you can see here the values are different. Nevertheless, we went on with this particular study. I'll get back to that in a minute. But what this study allowed us to do, because we'd got this line with GFP in it, we could fish out these GFP-positive cardiomyocytes from mixed populations in these directed differentiation, and then just do regular Western blotting. And what this showed us, so this ion channel with the mutation is called the hERG channel. This is the protein on the cell surface, and this is the intracellular protein. And basically what you can see, there's more intracellular protein than there is in the control line, which was for the first time when we did this study, this showed that this mutation was a trafficking mutation. So the protein doesn't get to the cell surface, therefore there are fewer ion channels that are functional on the surface of this cardiomyocyte. And this is why the people get sick, right? So this is why they have arrhythmias. So we can now look for drugs in the category that affect trafficking of proteins to see whether we can find something specific to fix it. So this kind of gives us a model. But because of that worrying thing, what was the difference now between the iPS cardiomyocytes and the ES cardiomyocytes? We then decided to go back to the literature. As I said, nothing better than doing a review. So I got one of the postdocs, Lucas Sala, to look in the literature and to find all of the papers in which the action potentials, the patch clamp, had been done in exactly the same way with the exact same fluids in the pipettes and everything the same. And he found 19 papers in which it had all been done the same. So then he looked at the absolute values they reported for their action potential duration. And you can see this whole range of values. So if you're looking at the isogenic pairs, for example, that they're the open versus the closed circles, so patient-derived and the isogenic controls, that's fine. You can see the difference you expect. But if you took a commercial line, which may be up here, you bought the cardiomyocytes, and your patient line was down here, then you're in trouble, because if you think this was a long QT, you say, I got it from a patient. But compared with the control commercial line, it would appear as a short QT. So you'd be totally confused. And we did this partly because somebody in a group in the US was doing exactly that experiment. He said, I can't measure long QT in my long QT line. It's shorter than my controls. I said, what's your control? Oh, well, I bought it. So it's good to know exactly what your baseline is, and this emphasizes the point, I hope, of isogenic pairs. So going back to our model, this is how you can actually use these cells to look for different compounds that may improve and shorten your QT interval. So we did this for several lines. You can see them here. One is a long QT 1 line, and this is a long QT 2. So we've been looking for allosteric compounds that can shorten this QT interval and see how it compares with the genetic rescue. And you can see these compounds here give basically the same sort of shortening as you see with genetic repair. So that's the kind of assay you want, and that's what you're looking for. And you can see the dose response curve here. This is the disease line, this is the treated, and you can see that. Again, you see this difference. So this is the disease line, and this is the control treated with drugs. So you'd think you'd still got the disease. So it's not the absolute values, it's the relative values that are important. So the importance of isogenic pairs may be now evident, but if you look at the literature, what you find is that many, many papers, so this is the number of papers doing this type of study, most of the papers still take an unrelated control as the comparator. There are a number of studies that take a related family member who's unaffected as a control, but there are still relatively few papers truly taking an isogenic control. And I expect, of course, this will increase. The easier CRISPR-Cas is becoming, then the more chances are that you have to do that. And when you're setting up a study, if working on a monogenetic diseases, we see in the literature now, or the referees' comments say, as a minor revision, compare with isogenic control. So that's a very sobering thought. That's a year's work for most people. Okay, so maturation is still an issue. So one of the things we decided to do was to see whether we could get anywhere doing metabolic maturation. And we used a very simple assay. We looked at the volume. We used a dye for mitochondria, which could give us a sort of reflection of the volume of mitochondria in the cell. And we know when cardiomyocytes form, they use an awful lot of energy, and they have a lot of mitochondria. And if we compared that with the level of GFP, driven by NKX 2.5, the reason for that is even though it's a transcription factor, it's a cytoplasmic GFP. So the ratio of mitochondrial to cell volume in cardiomyocytes goes up over time. And this is a dose response curve with a dye. So we can measure this ratio as a simple measure of the relative number of mitochondria in the cell. And you can see here, when we added a number of compounds, so T3 hormone, IGF-1, and dexamethasone, which are sort of candidates you would want to use, this ratio of mitochondrial to cell volume increases. And what we found was that this was reflected in the action potential. So we didn't do anything with the action potential. We just gave them stuff that would make them metabolically more mature. And I think you could try this for many other cell types. So basically, the action potential went up significantly. So we are actually close to adult values. It's about half the adult value here. But as I said, what we also wanted to do was to look at more parameters and using multi-parameter system. Now, I don't know whether this will work. Might do. But this is a measure, this is a single cardiomyocyte, and instead of keeping it on plastic, we've now kept it on a soft polymer substrate to de-stress it. So this is a single cell. It's supposed to be contracting, but it won't. But this is the calcium, so we load it with a calcium reporter. So this is a dye, Fura2. You can see the calcium going up and down. This is the cell contracting. But what we can also do is on that soft polymer substrate, we can put little beads in and track those as well. So what this allows us to do is actually measure the calcium transient. We can put in a voltage-sensitive dye and measure the action potential and the contraction simultaneously. So this is the action potential, this is the calcium, and this is the contraction, which follows the calcium wave. And if you add in a drug, so this is theoretically what should happen. And when we actually did this in a real live experiment, so this is only done with particular dyes and making movies. Of course, they're very fancy movies and a fast camera, but nevertheless. So this is the control in real life with these cells. This is a peak, and this is the action potential, calcium, and contraction. And if we add in a drug called digoxin, which is meant to increase the contraction of your heart, they give it to patients, it's from Foxglove, you can see here we get this massive increase in calcium and a massive increase in contraction. But we're measuring this simultaneously, not one behind the other. And this three-parameter system we're now using in the context of a CRACIT consortium, which is led by Chris Denning in Nottingham. And what we're doing is using different formats of these cells, so single cells, monolayers of cells. Thomas Eschenhagen's in this consortium, so he's using engineered heart tissues, using different formats. We're also using our microtissues that I'll get to. And we're comparing a single set of drugs. So we first did some standard drugs, about 12 compounds, which we knew what they were, and they would be tested, all of us identically, from the same batch that was supplied by GSK, and we got the outcome. And now we're testing a second set of drugs, 28 drugs, doing dose-response curves in each of our systems in a common cardiomyocyte, so everybody's using the same cardiomyocyte, everybody's using the same batch of drugs, everybody's diluting them even in the same batch of DMSO. Everything's identical, and our conclusions could be, well, what is the best cardiomyocyte to do these assays on, and which is the best assay? And the best assay is not necessarily the most accurate. We predict, because the engineered heart tissues are far more mature in terms of their features of the cardiomyocytes, but it may be the costs may be too high. You need 800,000 cardiomyocytes for an engineered heart tissue. You just perhaps need a few here, and in our microtissues you only need 30 or 40,000. So it depends. We'll see what the outcome of that is. But this maturation is quite important, because what you see in our metabolically mature conditions, you see this is a measure of the force of contraction measured on these single cells. You can see here the force of contraction goes up. And this allows us to measure a disease phenotype that we couldn't see without maturation. So this is a mutation in myosin-binding protein C3. So heart cells contract because they've got these sarcomeres. And this is a protein that's a component of the sarcomeres. We had a patient, a young boy, collapsed he's 14, playing football near the hospital, brought into the emergency, reanimated, and it turned out that he had a huge septum, a very large division in the chamber of his heart. And we figured out, or the geneticist figured out, he'd got this mutation. He didn't know. Turned out his father had it as well. And we were able to make iPS cells from these two individuals. And you can see here, their cardiomyocytes both had lower forces of contraction, even though the father had no symptoms. So this is the boy, and this is an unrelated control. We saw nothing when we didn't use metabolically mature conditions. There was no difference. So that shows you the value. Moving into more complex systems, we can make hearts on a chip. So we can make these soft polymers, we can turn them into microelectrode arrays and actually make them oscillate by a vacuum underneath, which we can make them oscillate at, say, 60 times a minute, 120, whatever heart rate we want. And we're making models of heart failure, using these kind of genetic mutations, but also myocardial infarction by depriving the cells of oxygen. So you can see this thing oscillating here. It's a bit like a contact lens going up and down, and we can pace it. And making vessels on chip, you can see this little lump here. This is blood flowing through a microfluidic chamber, and you can see on the downside of this little indentation, there's a thrombus forming. So using microfluidic systems, we can look at atherosclerosis, thrombosis. We can create blood-brain barriers if we get the right 3-cell types. We can look at vasculitis, which is inflammation of the vasculature, and maybe vascular dementia, which is one of the predominant forms of dementia. Not everything's Alzheimer. It looks like Alzheimer, but it isn't necessarily. And this is how we look at inflammation. So we look at leukocyte adhesion under flow. So this is one of these microfluidic chips, and you can put cells in them. The cells will flow over an endothelial layer. This is what it looks like. So this is our controls. So this is the profile of the different cells in terms of their adhesion molecules. So you can see this is HUVEC. So these different types of endothelial cells from iPS cells, CD31 or CD34 negative, look rather positive, look rather similar to HUVEC, and you can see these here for the other factors. And this is when we add TNF, which is an inflammatory factor. So this is the untreated controls. What you can't really see under here is a monolayer of endothelial cells derived from iPS cells, and these things moving across the top are leukocytes. So if we add TNF and increase inflammation, so what you can see here, a little bit slow, but what you can see here is there's cells sticking on here. These are the leukocytes sticking to the endothelial cells that have received TNF for inflammation. And if you quantify that, you can see it very clearly here. So this is the endothelial cells and HUVEC, and you can see in a quantitative way they react to inflammation. So this is really very cool. I'm getting near the end. So I just wanted to make one more point, and that was batch-to-batch variability, right? So we discussed this. And one of the things we've done is look very carefully at batch-to-batch variability. So we do this with these endothelial cells on something called EBiSC, so electrical cell substrate impedance sensing. You can do this with an epithelial layer, and you look at the resistance. So this is some data that comes out of this. So I told you there were CD31 positive endothelial cells and CD34 positive. CD34 positive form first, and then they turn into CD31 positive cells a little bit later. And what you see here is if we take the CD31 positive cells and we put them in this assay, it doesn't matter. In this case, they've been derived from fibroblasts or cells in urine. And this is the controls. You can see here, if we take batch 1, 2, or 3, these are all separate differentiations. The values we get out of this impedant assay are identical, right? But if we take the cells a couple of days earlier, when they're CD34 positive, so they have many properties of endothelial cells, look at the difference. They have not stabilized. So let's suppose these were disease lines. Just batch-to-batch variation would make it impossible for us to see a disease phenotype. So you've got to know how stable your readout is. We've done this with 50 cell lines now, and the values all come out to here if we use a CD31. There's been one exception, and it was way up here. It was completely wrong. So we could spot immediately that this was an abnormal behavior of a control. It was nothing to do with the disease. So as I said, we're interested in organs on chip. We're making brain, heart, and gut on a chip with a large consortium. This is making a brain cell, putting it into a heart chip, so a brain chip, integrating sensing, and also having flow of the blood vessels through this. So our next, and having electrical readouts, nanosensors, making hearts on a chip, a sort of ECG on this. I like these movies because it tells you what you might do in the future. And this is gut on a chip, putting the microbiome in it. And the microbiome determines also what's secreted by the gut cells. This could be a molecule that's important for controlling cardiovascular health or brain health. So we know, for example, there's an association between your microbiome and depression or cardiovascular disease. So we hope by making these individual chips, we'll make complex models, and when we link them up, we may be able to find the relationship between disease. Bit of fantasy. My last example is not from my lab, and it's not hard, but I think this is a very cool example of what IPS disease modeling can mean. Don't know whether it's going to work, but this is work from Kevin Egan. You likely know this work very well, but I really like this. What he did was to look at ALS. So ALS is a motor neuron disease, and there's very rare forms caused by a mutation SOD1 gene. And the SOD1 gene has no phenotype if it's mutated in mice, or no real phenotype. But he got a hold of a patient with an SOD1 mutation, corrected the gene, and in short, just did patch clamp electrophysiology to show that there was an imbalance in sodium potassium in those neurons from the ALS patient that was not evident in the control. Now that was quite interesting. He also showed an hyperexcitability of these neurons. So if you put a magnet next to an ALS patient, next to their head, their hand goes like this. They have these hyperexcitable neurons. And what he found, what he asked was, could he find a drug that would go through the blood-brain barrier that would correct the sodium-potassium imbalance? And there is an anti-epileptic used in the clinic, this one here, and he found that it corrected the sodium-potassium balance in these cells, and it corrected the hyperexcitability in the dish. But the clue of this paper was he took a second rare mutation, which is unrelated to SOD1 in mechanism, but the phenotype was the same. And he could correct the phenotype, not the genotype, with this drug. So he now has permission to go straight into a trial, no extra animal experiments, straight into a trial based on these IPS results. So he's taking 100 patients, as far as I understand, making IPS cells from them, looking at the effects of this drug on their motor neurons, and giving them this drug in the clinic. Yeah, so they'll see in 5 years' time, because all of the patients die within 5 years except Stephen Hawking. But they will be able to see in the outcome whether there's any correlation. So this is very cool. So the future of IPS in disease modeling and drug discovery, I think isogenic pairs with human disease-relevant mutations will be widely available, hopefully from the banks. We'll just call somebody in the future and sign a form. There will be documented informed consent, hopefully, genome sequence and donor medical history, including the drug responses. This is what we need. This is what the banks can mean to us. They could provide the missing link with genome-wide association studies through precision genetic engineering. We can now mutate SNPs, introduce variants upstream. If our phenotype readouts are good enough, we should be able to pick up these differences if we do the experiments right. So we cannot do these studies in mice. And drug combinations could be tested at different doses for effectiveness and toxicity in patients. They could be meaningful. And high-throughput bioassays, including organs-on-chip and 3D formats, will be available or should be developed to look for new drug treatments that delay the onset of symptoms. Reversing symptoms, once they've happened, is hard. Once the cell's gone, regenerative medicine's really hard. But if you can promote the survival of the cell that gets lost or treat the phenotype, then you may be there. So just to acknowledge the people in my group who've done all this work, we have an IPS core facility, and these are the junior PIs that are guiding the PhD students. These are our many collaborators and our funding bodies. And I just like to make a plug for stem cell reports, because we find this really important, this robustness. We ask for robust, so if you report a new protocol for doing X, Y, or Z, we require it to be tested on minimally 3 independent cell lines, and for disease modeling, 3 patients and 3 clones per disease, unless it's a very rare disease or unless you've got isogenic pairs. So just, you know, there's many papers that are not correct out there. So thanks very much.
That was terrific, Christine. Thank you so much. We're slightly over time, but perhaps time for a couple of questions. Actually, perhaps I could start. The modelling you've described for heart, I mean, are clearly very powerful. How far is this impinged in the pharmaceutical industry? Are those kind of screens being now used for, because you've shown some established drugs?
Yeah.
Are they being used now to develop novel drugs? Is that something that's going on?
Yes, so the interest in novel drugs, particularly repurposing, is growing. The industry is largely waiting on the sidelines still. They haven't taken the plunge, but the FDA has. So this cardiac tox as a replacement, so if you were looking for drugs that cause arrhythmia, the current guidelines say you have to express that ion channel in a hex cell or something and look at the effect of that drug on the hex cell, but it's turned out that that's not accurate. So the FDA has started what they call the CEPA initiative, and they will want, within a year or two, the effects of any new drug on the market to be looked at all ion channels. And the only way to do that is in an iPS cardiomyocyte. So there is a large consortium of small and large biotech screening a lot of compounds to satisfy the FDA that their requirements are real. And so that it certainly will have to be adopted in terms of cardiac tox.
Interesting, that's interesting. Further question? Okay, well, thank you very much.